So, right. So here, right, I shown to. So here, mass production. That is the what? Yeah. So the preparation appropriate media for a specific to the bacterial inoculant in 250 ml, 500 ml, 3 liters, and 5 liter. Conical flasks and autoclave sterilized at 121.5 degrees and 15 um, that is the pressure containing 50 minutes into 250 ml capacity of conical flask containing autoclave sterilized liquid media under the aseptic conditions. So place the conical flask under room temperature on the rotary shaker for 1 to 7 days, depending on the whether they are fast growing or slow growing. So observe the flask for the growth of the culture and estimate the population by dilution play technique. So which serve the starter culture. The content of these flasks usually attain a load of 10 to power of 5 to 10 to power of 6 cells is called as a mother culture or starter culture. These mother culture are further used for manipulation in large class for large scale production. So using the starter culture, inoculant the larger flask, 3 liters or 5 liters containing appropriate media. After obtaining growth in each flask, the above media has to be prepared in large quantities in the fermenter, sterilized well, so cooled and kept in ready for inoculation. So, so yes. So the media mm, in the fermenter has been inoculated with the long phase culture grown in 15-liter flask, usually 1 to 2 percentage inoculum is sufficient. However, inoculation can be done up to 5% depending on the growth of the culture in large flask. The cells has to be grown in fermenter by providing aeration uh, like glass, wool, cotton, acid, etc. By giving the continuous stirring, check the broth for the population of inoculated organism and contamination, if any, at the growth period. Harvest the cells with the population load of the 20 power of 9 cells. After inoculation period, ensure no fungal or any other bacterial contamination. It is not advisable to store the broth after fermentation for period longer than 24 hours, even at 4 degrees number of viable cells being to the decreases. Preparation of liquid broth culture. Preparation liquid media for the respective organisms. Distribute equal quantity is the big conical flask. Sterilize it is in autoclave for half of an hour. Hmm. After sterilization, inoculate each flask containing suitable broth with the mother culture in 1 is to 15 proportions aseptically. Keep the flask on rotary shaker for 96 to 120 hours until viable count reaches to 10%. So the broth culture became thick in consistency. This broth culture with population of 24 or 9 cells stored in 4 degrees temperature in the refrigerator should not be stored for the more than 24 hours. So preparation of carrier preparation of inoculant, that is the mixing and curing or mm, so here the use of ideal carrier material is necessary in the production of good quality biofertilizer, peat soil, dignate, vermiculture, charcoal, fresh mud, and farmyard manure and soil mixture can be used as a carrier material. So, so the preparation of carrier material. Wait. So preparation of carrier material. The carrier material, that is the lignite or peat, is the generally used as the carrier material in the preparation of biofertilizers. Crush and powder is the carrier material, right? Okay. Right. So, mm, so crush and powder the carrier material so as the cost during the 200 to 300 microns. Then utilize the pH carrier material by adding 1% of calcium carbonate and sterilize the neutralized carrier material into the autoclave. Hmm. The sterilized and neutralized lignite or peat is mixed with the bacterial broth culture in the galvanized trays and mix well. So about the 1 part of weight of broth is required to 2 parts per carrier. Final moisture content is from 40 to 50 depending upon the quality of the carriers. So counting on the or maturation. Maximum liquid or broth culture is thoroughly with the autoclave sterilize the carrier material, lignite or peat powder in 1:2 proportion in the galvanized trays. So then kept for the curing can be done by spreading the inoculation on the clean floor polythene sheet by keeping the open shallow. So here 1 to 2 days at room temperature. So filling and packing the quality checking storage. Filling and packing after curing, fill the seaweed powder in polythene bags of low density grade with the zero point 0.5 to 0.75 mm thickness, leaving 2 by 3 space open aeration for the bacteria. Bag is weighed for the designed quantity. Then the bag is packed by the sealing. The polythene bag is used for filling the microbial inoculant. Should be printed with the following information. So name of the inoculant, stain product, staining name, and direction used for use. Name of the crop with the which is the recommended method of inoculant. Name of the manufacturer, date of the manufacturer, date of expiry, price. So quality checking. The viable count in the carrier-based inoculant has been checked by the dilution plate method at the time of manufacture. The viable cells count in the carrier-based inoculant should be maintained as per the ASI specifications. The storage in the inoculant has been stored by the manufacturer in a cool and dry place away from the direct heat or direct sunlight, preferably at the room temperature. For long survival of the microorganism, the bags can be stored in the cold storage condition at 4 degrees. The population of inoculant in the carrier inoculant packing may be determine at 15 days interval, there should be more than um, so economic cost and availability. So, so nationalized banks have started their high-tech agriculture program, providing the loans and motivation the entrepreneurs to start their own production. So, so with the object to give the overall economics of the biofertilizer production and scales in the approximate estimation is prepared. Total estimation for starting a biofertilizer production unit with the capacity of 150 metric tons. So here that is a particular expenditure. That is there. So now coming to this here, cost of the name of the biofertilizer here, azospirillum cost of biofertilizer here, 40 rupees per kg, phosphobacteria and 40 rupees per kg, rhizobacterium, azotobacter, the PAM fungi, vascular or vascular mycorrhiza, 30 rupees per kg. Now, that is the unit 6, biofertilizers, sustainable agriculture. That is, yeah, we discuss introduction. The conventional agriculture plays a significant role. So demand of the growing human population, which has also led to the increasing dependence for the chemical fertilizers and pesticides and their exploitation causes air and groundwater pollution by eutrophication of water bodies. The exploitation of beneficial microbes as a biofertilizer has become the paramount importance in agriculture sector for their potential role in the food safety and sustainable agriculture production and biosafety program. Uh, the biofertilizers mediate crops information, crops functional traits such as the plant growth and productivity, nutrient profile, plant defense and protection with the special emphasis to its function to trigger various growth and defense related genes in the signaling network for cellular pathways to cause the cellular response and thereby crop improvement. In this regard, the recent efforts have been channelized more towards the production of nutrient rich high quality food for sustainable agriculture to ensure biosafety. So sustainable agriculture is the product um, and efficient way to produce safe, high quality agricultural products in the way they protect and improve the natural, environmental, and socio-economic conditions for farmer, their employee, and local communities and safeguards. So mechanism of action. So mycorrhizal functional association with the roots of higher plants serve as a model system to understand the mechanism behind the stimulation of growth in the root cells as well as result of mycorrhizal inhabitation. So 15th genes of regulate during the symbiosis were identified to to Lexus transport in the that is the Laceria bipolar. So the bioactive compounds called make factor similar to inverted factor of rhizobium are suggested to be secreted by the mycorrhiza and rhizobium and that is perceived by the host roots for the activation of signal transduction pathway or common symbiosis pathway. Many diseases resistance genes that work via jasmonate, ethylene, and signaling as well as osmotic regulation via proline synthesis gene were differentially expressed with the bacillus stubulus. When the biofertilizers are applied as seed or soil inoculant, they are multiply and participate in the nutrient cycling and benefit crop productivity. In general, 60 to 90% of total applied fertilizers is lot and the remaining 10 to 40% is. In this regard, microbial inoculants have paramount significance in the integrated nutrient management system to sustain agriculture productivity and the rhizosphere, which is the narrow zone of the soil surrounding plant roots, can comprise up to 10 to the power of 11 microbial cells per gram of food and above 35 prokaryotic species that in the general and improve the plant productivity. The collective genome of rhizosphere microbial community enveloping plant roots is referred as microbiome. Those interactions determine crop health in the natural agroecosystem by providing the numerous services to croplands like organic matter decomposition, nutrient acquisition, water absorption, nutrient recycling, weed control, and biocontrol. PGPR produce plant growth hormone, indolestic acid, in turn to introduce uh, the second messenger to trigger complex signaling network lead to improve the root growth and development process. So nitrogen fixing bacteria can fix atmospheric nitrogen by the induction of nest genes that takes place under the low concentration of nitrogen and oxygen in the atmosphere. So various differentially expressed genes induced by bacteria with Bacillus stabilis and the thionine family protein and origin gamma chain precursor stress association proteins. So among the PGPR species, Azospirillum was reported to secret growth hormone such as gibberellin, ethylene, and auxin. Some plants associate bacteria can also induce the phytohormone synthesis. For example, indole pole pine with the inoculated with Phenibacillus that level of IA in the roots. Rhizobium ambassadors are found in the synthesized IA at the different cultural conditions such as pH temperatures and in the presence of agro-waste as substrate. Ethylene, unlike other phytohormones, is responsible for the inhibition of growth of the dicot plant. Earlier researchers found the PGFR could enhance the growth of plant by suppressing the expression of ethylene. So interestingly, the model was suggested in the it was shown that the ethylene system from the one amino propylene carboxylate is the immediate precursor of ethylene, which is the hydrolyzed by the bacterial uh, amino cyclopropane one carboxylate lead to nitrogen and carbon source. It is also one of the mechanism of the induction of condition sustainable growth. So next one is the mechanisms. Soil microorganisms that is the inhibiting around on the root surface and are directly or indirectly involved in promoting plant growth and development via production and secretion of various regulatory chemicals in the vicinity of rhizosphere. Generally, the soil microorganisms uh, facilitate the plant growth directly by either assessing the resource acquisition, phosphorus, and essential minerals. So, different bacteria genera are vital components of soils. They are involved in the various biotic activity. Check, check So, the plant growth to mobilize the movements in the soil, producing numerous plant growth regulators. So, protecting plants from the phytopathogen by the controlling or inhibiting them, improving the soil structure, and by remediating the polluted soil by sequencing toxic heavy metal species and degrading xenomic compounds. So therefore, these rhizosphere are these organisms that are dominated by deriving forces, recycling the soil nutrient, and consequently they are the crucial for soil fertility. So mechanism of plant growth promoter, that is the PGPR. So PGPR means plant growth promoting bacteria. That one we call as the PGPR. So here PGPR bacteria is shown to that one. So PGPR bacteria. So PGPR, that is the mediated plant growth promotion occur by the alternation of the whole microbial community in rhizosphere, which through the production of various substances. So although the mechanism of the rhizobacteria-mediated plant growth promotion are not completely identified, generally PGPR promote the plant growth directly by either facilitating resource acquisition or modulating the plant hormone level or indirectly by decreasing the inhibitory effect of various pathogens on the plant growth and development in the form of the biocontrol agent. Direct mechanism. So the atmospheric nitrogen is converted into plant usage from the biological nitrogen fixation, which changes the nitrogen to ammonia by the nitrogen microorganism using a complex enzyme system known as the nitrogenase. So nitrogen fixing microorganisms are widely distributed in the nature. There is a furthermore biological nitrogen fixing will represent an economically beneficial and environmentally sound alternate to chemical fertilizers. These organisms are generally categorized as symbiotic nitrogen fixing bacteria And the non-legumes, that is the frankia and non-symbiotic living organisms, that is the Anabina, azotobacteria, azospirillum. These are the uh, examples of the non-symbiotic trees. So the plant growth promoting rhizobacteria can fix the nitrogen non-leguminous plants, also called as the diazotrophs, are capable. So that is the dinitrogen reductase, which is the improved iron protein, and dinitrogenase, which has metal cofactor. Dinitrogenase reductase to provide electrons with high reducing power, while dinitrogenase using the electrons to reduce. Based on the metal cofactor, three different nitrogen fixing systems have been identified. That is nitrogenase, V nitrogenase, and Fe nitrogenase. Mm. So genes for nitrogen fixing called as the NIF genes are found in both symbiotic or free living system. So nitrogenase genes or NIF genes include the structural genes. Genes are involved in the activation of iron protein, iron molybdenum, cofactors, biosynthesis, electron donation, and regulatory genes all are the required for the synthesis and function of enzyme. In the diazotrophs, the NIF genes are also especially found in the cluster of around the 20 to 24 with the seven operation including the different portions. The molybdenum nitrogenase enzyme complex has two component protein encountered by the NIF genes decay and the NIF genes in decay. So phosphate solubilization. The plants absorb phosphate only in the two soluble forms, that is the monobasic and diabasic. So of the various phosphate solubilization mechanisms inhabiting the rhizosphere, phosphorus solubilizing bacteria are considered as promotion. So the typically the whole solubilization of inorganic phosphorus occur in the consequence of the action of low as consequence of low molecular weight to organic acid with the synthesized so molecular bacteria, right? Sorry, innovative, innovative. So, so I'm shown to another diagram. మేడం అనుకోకుండా సో Sidero, Sideropass, Siderophore production. All the microorganisms known with the exception of the certain lactobacilli is essentially required iron. In the aerobic environment, iron occur particularly as the iron and is likely to be found the insoluble. చెప్తాం నువ్వు ఎందుకు సో phytochrome production, so microbial synthesis of phytohormone auxin, that is the indolestic acid, and has been known as for long time. It is required. It is reported that 80% of microorganisms isolated for the rhizosphere of various crops possess of ability to synthesize and release the auxins and secondary metabolites. Generally, IAA secreted by Rhizobacteria interferes with the many plants' developmental process because the endogenous pool of plant IAA may be altered by the acquisition of IAA that has been secreted by the soil bacteria. So indolestic acid plays a very important role in the Rhizobacteria plant interactor. Regulating the IAA as signaling is associated with the plant defense mechanism against the number of phytopathogenic bacteria as evidence in the enhanced acceptability of plant to be bacteria pathogen by excess applications of IAA. So generally IAA affect the plant cell division, extension and differentiation stimulates seed and tubular germination, increase the rate of the xylem and root development, control process of vegetative growth, initiative, lateral and advantageous root formation, mediated responses to light, gravity, and fluorescence effect, the photosynthesis, pigment formation, biosynthesis of various metabolites, and resistance to stressful conditions. IA produced by the rhizobacteria likely interfere in the above physiological process of plant by the changing the plant oxygen flow. Um, an important that is the molecule that alters the level of IAA synthesis is amino acid tryptophan, identified as the main precursor of IAA and thus plays the role in modulating the level of IAA. So biosynthesis strongly the tryptophan stimulate IAA production while anthralate, a precursor of tryptophan, reduces IAA synthesis by these mechanisms. IA biosynthesis is fine termed because the tryptophan inhibits uh, anthralate formation by the negative feedback regulation on the anthra, anthranilate synthesis, resulting in the induction IA production. So IA producing bacteria, the genera include the Agrobacterium, Pseudomonas, and that is the Azospirillum, Klebsiella, and Enterobacteria, Cyanobacteria. So limitations, and what are the benefits of these Agrobacterium means what are the benefits? The important, the innovative view of from the production of attract growth to demand of biological based on organic fertilizer, exclusive of the alternate to agrochemical. In agriculture, that encouragement of for the alternate means of soil fertilization, relief on organic inputs to improve the nutrient supply and conserve the field management. In agriculture, the organic farming, that is the Chagli that encourages alternate means of the soil fertilization that not only ensure the food safety but also add to the biodiversity of soil. So organic farming phase is the rapidly growing in the locality, but in organic farming has been around wherever the organic farming in the pathway that is the leads to live in the harmony with the nature, mostly depending upon the natural microflora of the soil, which is constant. The advantage of organic farming. So, organic farming helps in the maintaining the environmental health and reduces population. It ensures the optimum utilization of resources for the short-term benefits and conserve them for the future generations. It helps to in improving the soils and physiological properties and ultimately agricultural productivity. So organic farming ensure the optimum productivity with the no demand for costly input. It believes in sustainable productivity with any loss in the future fertility, causing no adverse effect to ecosystem. Besides this, farming they promotes multiple cropping and reduce the risk of crop failures. So organic farming is the elimination the risk of human and animal hazards by eliminating the chances of chemical residues products. This farming the nutritionally rich and balanced. So challenges to improve the crop production. So agriculture is essential to human survival and social development with the worldwide human population growth and economic development. So increasing the development for agricultural products has placed that is a sustainable. pressure on agriculture and natural resources. This is the term has caused environmental pollution and ecological degradation. So agricultural sustainability has become a crucial problem that is the central to the sustainable development of complex socio-economic natural system. A major focus in the coming decades would be the on the safe and eco-friendly method by exploiting the beneficial microorganisms in the sustainable crop production. So different kinds of soil microorganisms belong to several tags of the bacteria, fungi, and possibly protozoa kingdoms, colonizing the rhizosphere or the plant tissues and promoting plant growth can be utilized for the production of microbial based fertilization. So the agriculturally useful microbial population cover plant growth, promoting rhizobacteria, cyanobacteria, mycorrhiza, plant diseases, suppressive, beneficial bacteria, stress tolerant endophytes, and biodegrading microorganisms. So coming to the here biofertilizers, low cost technology, that is the seventh unit is there. Um, in the So low cost technology means how the biofertilizer replaced in the very low cost here. So the preparation of biofertilizers using the various waste, including the livestock and poultry waste and agricultural waste as the main raw materials. There is a fecal or waste do not only create the economic benefits for the enterprises but also provide the great contributes to human environmental engineering. So after safe treatment of fermentation, these raw material are made into biofertilizer using the appropriate microorganisms. So this production system is not only turn the waste into but also protect the environment. So the several kind of agro waste will be that is to good and um, carrier material for the microbial inoculants. The biodegradable such as material, simple sugar, providing energy source for the heterotrophic to microorganisms such as the phosphate stabilizing and nitrogen fixing bacteria. So agriculture waste means agro waste as defined as waste which is produced from the various agricultural activity, which include the manure and bending plant stalks, hulk, hulls, leaves, and vegetable matter, whatever it may be. The agriculture usually produced through the farming activity. In the farming situation, agro waste is often unless and will be discarded. The accumulation of agro waste may be cause health and safety, environmental and aesthetic concern, which is a problem to with the disposal. Okay, so now this one is the thank you for the